fMRI.
Great. All right, cool. Thank you for the introduction. And so while I'm waiting for my slides, I'd like to thank Amir for organizing such a great educational session today. I'm really happy to be here. Thank you again for inviting me. And so I've been asked to talk about preprocessing. There we go. I've been asked to talk about preprocessing and analysis strategies for laminar fMRI. And so why a talk on this topic? Well, as we've seen, there's been growing interest in laminar fMRI in humans. It's known from invasive animal studies that different cortical layers have distinct functional roles, and therefore, a laminar fMRI may be the key to understanding functional architecture and brain circuitry in humans. However, it's very challenging from a data analysis standpoint. We'll see that conventional preprocessing and analysis techniques really aren't appropriate for laminar fMRI. This is partly because we're really at the edge of being able to detect fMRI changes in layers with our current resolutions. Today, we can't resolve layers either functionally or anatomically with our current voxel sizes. And also, it's important that there's a major interpretational challenges when it comes to laminar fMRI. We've heard about this many times this morning. There's a lot of hemodynamic coupling and spreading across layers, and therefore it's very difficult to infer the underlying pattern of neural activity from the fMRI signals we measure across layers. And so with this brief introduction, I'd like to give an outline of this presentation. I'll start by talking about anatomical reference data for high-resolution fMRI and ask how can we delineate accurately the cortical boundaries in our fMRI data. Next, I'll talk a little bit about preprocessing for intracortical fMRI and ask how do we preserve the high spatial temporal resolution in our imaging data. Next, I'll talk about analyzing fMRI data across both cortical depths and cortical layers. These are kind of related concepts but different, and I'll define them both in a few minutes. And there I'll ask how do we localize activation to individual layers with our current voxel sizes. And finally, I think this talk wouldn't be complete without a little bit of a discussion of the cortical depth dependence of the hemodynamic response. There I'll ask how non-uniform is the hemodynamic response across layers. So first, I think I need to say a few words about anatomical reference data, because again, in laminar fMRI, we can't resolve or we can't detect individual layers. We need to estimate the layers from the cortical boundaries, and therefore it's really important to get accurate segmentations of the cortical gray matter. And so obviously we've been moving to smaller voxel sizes that we heard this morning, both in terms of our functional and anatomical data. We wanted to revisit the assumption that a 1 millimeter isotropic, say, MP range acquisition was sufficient for accurately delineating the cortical boundaries in our human data. So we sat down with Bruce Fischel and the FreeServer team to make new modifications to FreeServer to allow ReconAll to be run directly on sub-millimeter data. So here are some examples of this new Recon stream on some 750 micron isotropic MP range data acquired at 7 Tesla. So with this higher resolution, we wanted to ask whether or not there'd be any advantages to going to higher resolution in terms of the placement of the surfaces. So here we looked across 40 subjects acquired at 7 Tesla. We compared surfaces reconstructed from 750 micron isotropic data with surfaces reconstructed from our standard 1 millimeter data. What we saw based on inspection is that the surfaces reconstructed from higher resolution data indeed were more accurate in terms of their placement. We also saw pretty consistently across subjects that in the surfaces reconstructed from the high resolution data, the cortex was mostly thinner in most of the cortical regions. However, there are a couple of regions in which we found that the cortical thickness estimates were thicker, including in areas like V1, S1, M1. This is also, V1, S1, and A1. This is also seen by Matt Glasser. These regions are more heavily myelinated, also very thin. So we're seeing that there are many advantages to using higher resolution data. We see that those advantages do vary across different regions. However, overall, we are seeing that we do see more accuracy in the surface reconstructions with these high resolution data with this new processing stream. So next, the question is, as we go to higher resolutions, of course, we're going to be sacrificing SNR. And in some cases, this push to higher resolution can be counterproductive. So just to demonstrate this, here's some 600 micron data acquired at 3 Tesla. We ran this through our new FreeServer Recon stream. The surfaces looked okay, but there are a few places where we really weren't happy with the accuracy. And at first, we weren't sure if this was due to the higher resolution in the data or due to the low SNR in these high resolution data. So to address this, we averaged together 6 of these 600 micron acquisitions to bring up the SNR, and there we were much more happy with the accuracy of the surface placement. So what this tells us is that, it seems that the errors we were seeing before were really due to lower SNR in these high resolution data. And so the point is, as many of you go to higher resolution anatomical acquisitions to get higher accuracy, it's really important to be mindful of losses of SNR. In some cases, it may be more advantageous to go into a larger voxel size in order to get sufficient SNR to get high surface reconstruction accuracy. So as we heard this morning, there's been a lot of advances towards improving the quality of our API data, mostly for fMRI. And so we're asking, is it possible that API has sufficient quality to be used for anatomical imaging as well? So here's an example of some of our high-resolution T1 mapping with an API readout at 7 Tesla. This is a 1 millimeter isotropic whole brain acquisition. Very simple pulse sequence. What we do is we play a non-selective inversion pulse. We read out all the slices, so each slice gets a different inversion time. We repeat this, playing a second non-selective inversion pulse, read out the slices a second time, but this time we permute the order. So during the second inversion recovery, every slice sees a new TI. Now, if we repeat this enough times, in this case we did 24 contrasts in about 3 minutes, each slice sampled 24 distinct inversion times with which we were able to fit T1 recovery to each EPI voxel in order to generate a quantitative T1 map with an EPI readout. With this quantitative T1 map, we can then synthesize T1 weighted images, such as this one with MP range-like contrast, which we can then pump through FreeSurfer to generate automatic cortical surface reconstructions and parcellations directly from 70 EPI data. I just want to point out that this general idea of using EPI as an anatomical reference has been seen by many different groups. We heard about this morning, both in the context of 2D EPI and 3D EPI. And just for fun, we wanted to see if we could play this trick at 3T as well. This is a 1 millimeter isotropic whole brain acquisition at 3 Tesla, again showing that we have enough SNR for this idea at 3T as well. This is our one image where we required about 7.5 minutes. I think we could do this faster. And so the question is now, why bother with this? Why bother generating surfaces directly from EPI data? Now, I wouldn't advocate using this for morphometric analyses. We know that the cortical thickness will be affected by distortions in the EPI data, but that's kind of the point. The idea here is that now that we have enough quality in the EPI data, we can generate anatomical reference data using the exact same EPI readout that we use for our functional data, so that the anatomical reference and the functional data are exactly distortion matched. This gets around, I think, one of the biggest problems with fMRI analysis is to get accurate alignment of the functional data to the anatomical reference. This becomes more and more important as we push to higher resolutions in order to achieve sub-millimeter accuracy. So I think this idea of generating cortical surface reconstructions from anatomical reference data that's exactly distortion matched to the fMRI data, I think is a good strategy. And now, so as David mentioned this morning, if we do want to remove the distortion from the EPI data directly, we need to estimate a B0 map, which as many of you know is very challenging to do. Also, most of the distortion in EPI is along the low bandwidth direction, which as you know is the phase encoding direction. But as we go to thinner and thinner slices and smaller voxel sizes, we're starting to see that the bandwidth in the slice select direction is also starting to become somewhat low. So we are seeing distortions along the slice select direction as well. So just to demonstrate this, we implemented a pulse sequence very similar to a sequence that many of you have used, which you reverse the polarity of the phase encoding readout. Here we've alternated the slice select gradient polarity just to see how much distortion there is along the slice select direction. So if we compare the images from a positive readout to a negative readout, positive, negative, you can see how much displacement there are of the voxels in the slice select direction. So what this means is that there is appreciable distortion in this direction. If you do try to estimate a B0 map to correct for the distortions in the EPI data, it's important to take these distortions into account, as well as the distortions along the phase encoding direction. But again, if we acquire an anatomical reference data that's exactly distortion matched to the fMRI, these distortions are not something that we need to be concerned with. So another idea is that, of course, if we have our B0 map, we can try to perform EPI and warping in order to remove the distortions from the functional data so that they align to the anatomical data. But of course, EPI and warping causes resolution losses due to interpolation. So here's a map showing the resolution losses intrinsic to the unwarping process. This is EPI data distortion corrected with standard field map-based correction. So you can see that the pattern of spatial resolution losses in these data look very similar to the B0 map itself. And I didn't use the audience response system, but a fabulous no prize goes to any audience member who can explain why this spatial resolution loss looks so similar to the B0 map. So again, on warping, the image requires interpolation, which causes resolution losses. They're also going to be a function of the voxel size. Again, voxel shifting on the image grid requires resampling and therefore blurs the data. So this is something we need to be mindful of if we want to perform distortion correction on the EPI data. Again, if we acquire anatomical reference data that's exactly distortion matched to the fMRI, this is not a concern. And with that, I'd like to move on to talk a little bit about preprocessing for intercortical fMRI. I know there's many different sources of resolution loss that could affect the ultimate resolution in our data. I showed you before how B0 distortion correction can cause resolution losses. Also, gradient nonlinearities have significant distortions. These distortions are a function of the gradient coil. They're also a function of the head position within the gradient coil. So we need to be mindful of these when we're comparing data across coils, across sessions, or even when the head shifts within a session. So here's a map of the resolution loss with one particular head gradient coil, body gradient coil. You can see that the pattern here is very different from the pattern I showed you in a previous slide, corresponding to the resolution losses that we see with B0 distortion correction. In this example, you see there's not much resolution loss at the top of the head near a motor cortex or somatosensory cortex. We're seeing much more resolution losses around the center of the brain, around V1 and maybe the posterior cingulate. So any form of interpolation is going to cause resolution losses with different spatial patterns. Here's a resolution loss attributed to a motion correction with a simple head nodding by 2 degrees. We see for a distinct pattern of resolution loss, head shaking by 2 degrees gives a different pattern of resolution losses. The point here, again, is that the interpolation causes resolution losses that are spatially varying. Now, the conventional approach is to apply each of our transformations individually. So, for example, if we have a motion correction transformation, EPI distortion correction and registration, followed by a gradient nonlinearity correction, these 3 consecutive steps of resampling give rise to a high degree of blurring. However, because the patterns of blurring in each of these steps is somewhat different, the blurring pattern tends to be spatially uniform. Now, this is what we see with a bilinear interpolation, sort of a low-order interpolation. We can mitigate this somewhat by going to a higher-order interpolant, such as spline interpolation, which still gives a high degree of blurring, which comes less spatially uniform. So I think a better approach that's been advocated in the Human Connectome project in a paper by Matt Glasser that I was involved with a few years back kind of advocates composing the transformations in order to minimize the number of interpolation steps applied to the data. So in this case, if we compose the transformations and perform a single resampling, there's less blurring. However, it does become somewhat spatially non-uniform. However, it's really straightforward to estimate the pattern of resolution losses if you know the transformations. So the idea here, again, is that we need to be mindful of these spatial resolution losses because in laminar fMRI, we're really at the edge of being able to detect these signal changes across layers, and therefore this can be another source of bias. So that's all I want to say about preprocessing. Next, analyzing fMRI data across cortical depths and cortical layers. So the very first step in laminar fMRI analysis is to simply sample the fMRI signals across cortical depths. And so therefore, we need to be mindful of the different levels of intercortical vascular anatomy in the cortex. Here are the vessels shown in the cast of macaque V1. Of course, we have these large PO veins that drain deoxygenated blood from large cortical veins lying along the PO surface. We also have these ascending venules that penetrate perpendicularly into the cortex. These are especially important in the context of laminar fMRI because they can be a source of a coupling of the fMRI signals across layers. And I'll be talking about that a little bit more in a few slides. And of course, within the parenchyma of this randomly oriented capillary bed. So we have to be mindful of the fact that fMRI signals across layers sample from different levels of the vascular hierarchy, and therefore we are expecting to see different couplings and different contributions of the vasculature as a function of cortical depth. So in order to perform this first step, we perform a cortical depth analysis. Here's an example of what I like to think of as a surface-based cortical depth analysis, in which we start by identifying the gray-white boundary. We reconstruct a surface there and evolve that mesh out to the PO surface, saving intermediate surfaces at various cortical depths. In this case, I looked at 9 intermediate or intercortical surfaces. I'm showing the cross-sections of these surfaces overlaid on the ambirage on the left there. So the idea behind this analysis is to simply identify our fMRI voxels to intersect each of these intercortical surfaces. So in a surface-based cortical depth analysis, we generate 1 cortical surface model per depth, and these surfaces in this example are placed at a set of equally spaced relative depths. Here I'm showing the 10%, 50%, 90% depth relative to the total cortical thickness. So this analysis enables a cortical depth analysis over large expanses of the folded cortex. So just to demonstrate, this analysis technique, we wanted to look at the cortical depth dependence of spatial accuracy, in this case in gradient echo bold. So here we designed a visual stimulus that would give us a resolution pattern, a known pattern of neural activation in the primary visual cortex in the shape of this letter M, just for fun. We sampled it across depths, and here are the activation maps that we could see with this cortical depth dependent analysis. So we saw that the spatial pattern of the activation of the letter M was pretty good as we sampled near to the white matter, which is away from those large PO vessels. But as we progressively sampled closer and closer to the PO surface, we saw that the spatial fidelity of this pattern broke down systematically, but I think in an interesting way. Here's a fun artifact here. It looks like there might be a PO vessel that's cutting off the top corner of this M. So as Amir had said, I think the point spread function is a very useful tool to summarize the resolution in an acquisition, but as he also said, in a given spot, the resolution doesn't seem to be well characterized by a zero-main point spread function. That's partly because our voxels are becoming so small, they're really sensitive to the local details of the vascular anatomy. So we can also use this approach to look how signal changes vary across cortical depths. We saw that in this study that the percent signal change near the white matter was about 1.5, whereas the percent signal changes that we measured near to the PO surface, which again are reflecting deoxygenation changes throughout the entire cortex, were about twice that, about 3%. So we see the steady increase in the sensitivity of the signal as we sample closer to the PO surface. We also looked at the spatial variability of the percent signal change, and we saw was that near to the white matter, we saw pretty much the same kind of low, small percent signal changes everywhere. But when we sampled near to the peel surface, we saw both big percent signal changes and small percent signal changes, which we think are reflecting the spatial heterogeneity of vessel sizes on the surface. At the peel surface, we have both big vessels and small vessels. So you have capillaries at the peel surface, and these big vessels are somewhat sparse. So we think that this broad distribution of vessel sizes may be the cause of the spatial variability that I'm showing here on the curve, on the red curve on the right. So what this is telling us is that these bold cortical depth profiles vary a lot over space. They vary very different from one location to the next, and the variability is most pronounced when sampling near to the peel surface, for gradient echo bold at least. These also show us that the profiles reflect different vascular contributions to the bold signal across cortical depths. So one issue that we need to be mindful of when using cortical surface-based depth analysis is the irregularity of the surface meshes. Both the vertex density varies both with the depth and curvature. So as I mentioned, we tend to start with a surface from the white matter. We generate a cortical surface reconstruction, propagate that mesh out to the peel surface. This propagation can lend itself to seeing a bunching of the vertices. This is shown recently in a nice paper by Kendrick Kay, in which he showed that with the free surfer method, there can be a bunching, a higher density of vertices, for example, in sulci at the peel surface. Now, this bunching can cause, when the fMRI data are projected onto the meshes, can be the cause of sampling biases. So there's a couple of ways in which we can correct for this. We can perform a vertex density correction. We can also use a more computationally intensive technique, which is really severely upsample the surfaces. But there's another approach. This is recent work from Valent and Kemper, in which he also looked at the regularity of the distances between neighboring vertices on the surface, and how they can vary across the folds. So the solution proposed and implemented in FreeSurfer is to replace this triangular mesh with a lattice or grid, or what I'd like to call a square quadrilateral mesh, where the density is uniform. So I think we'll be hearing more about that in the next presentation. So while these surface-based cortical depth analyses are really useful for visualizing the columnar patterns across the cortex and also across depths, there are some problems when we use these techniques for generating cortical laminar profiles. So the idea here is that if we consider our voxel grid superimposed on top of the folded cortex, we can see that any given voxel might be projected onto multiple surfaces, and this can be the cause of spreading of the signal across depths. So we can simply do away with these intercortical surfaces, calculate each voxel centroid in 3D, and then calculate the distance of that voxel centroid to the bounding surfaces of the cortex in order to assign to each voxel a cortical depth. So here's the differences that we see between so-called surface-based cortical depth profiling versus the proposed voxel-based cortical depth profiling. This is a fun example in which we're looking at physiological noise across depths. So in this plot here, just draw your attention to the blue curve. This corresponds to the percentage of variance explained in resting state bold with the retro-I-core regressor, which contains contributions from both cardiac and respiratory noise. We can see that the amount of physiological noise just very slowly increases in the surface-based cortical depth profile, very slowly increases as we sample closer and closer to the peel surface. However, if we consider the same data using a voxel-based cortical depth profile, we can see that the curve becomes much more L-shaped because there's less smearing along the depth dimension. We can better see that there's a transition between regions in which the voxels are sort of more dominated by noise from the white matter versus voxels being more dominated by noise from the CSF. So the idea here is that while there are some advantages to the surface-based cortical depth analysis, surface-based cortical depth profiling can smear activation across depths, and therefore to preserve resolution, I think it makes a lot of sense to use this voxel-based depth assignment. Now, we're doing something kind of strange in laminar fMRI. Many of you may be thinking that we're looking at these cortical depth profiles across cortex that's 2 to 3 millimeters thick. And typically, our voxels are about 1 millimeter in size or maybe barely sub-millimeter. So how do we do this? We do this by generating depth profiles by pooling voxels across an ROI. So this is nicely demonstrated by Peter Koopmans. So what Peter did was he looked at some profiles across cortical depths. In this case, it's in an example MP range. So first, he looked at the intensities across this green contour in the MP range, and he sampled it, I'm not sure how many, like 100 different depths. And what he saw was that if you looked at the intensities across this profile that was oriented parallel to the voxel grid, he saw that there's about 6 or 7 unique intensities. Okay, and then he considered the profile along this red contour shown on the bottom there, and he also sampled the MP range across the depths, And he saw that, again, there's about, in this case where the profile was oblique to the voxel grid, he saw that there's about 6 or 7 or maybe 8 unique intensities. But you can see that the transitions between these unique intensities occurred at different cortical depths across these two profiles. So you can see where this is going. If he pulls across many different profiles, in this case he averaged the profiles across 25 different locations, you can see that this gives a much sort of better approximation to the smooth intensity variation across the cortex. Now, I like to think of this as being somewhat akin to what we do often in event-related fMRI, when we're trying to estimate the hemodynamic response function at a higher temporal resolution than our temporal sampling interval. We do this by jittering the onset times of the stimuli and acquiring many trials in order to resolve this at higher resolution. It's kind of akin to what we're doing here in laminar depth profiling. Now, this assumes that all voxels within the ROI have a similar intensity or fMRI activation across layers. This is a pretty strong assumption, and the natural inclination with this assumption is to assume an ROI that's pretty much as small as possible. But there are some problems when we do this. This can introduce sampling biases across depths. So if you consider a very small ROI in the visual cortex, it might be hard to see. There it is there. And we look at all the voxels sampled within this ROI, we can see in this histogram that not all depths are sampled equally. In this example, we see a lot more voxels are sampling from the peel surface, and also in this example, there seem to be a lot of voxels sampling from the white matter surface. What that means is that any profile that we generate across this ROI is likely to be biased by the signal contributions from these two locations. So what can we do? We can make the ROI a little bit bigger. In this case, if you look at the histogram again, it looks to be a little bit more uniform, still not perfect. If we go to a larger ROI, which takes up about half of V1, we see that the distribution becomes much more flat, indicating we have minimal sampling bias across depths. So this is a trade-off in defining ROIs for laminar profiling. On the one hand, we want the ROIs to be as small as possible, so the assumption that all of the voxels at any given cortical depth have the same activation within the ROI. We also want the ROI to be large enough to avoid these depth sampling bias effects. Now, it's one challenge in estimating the positions of layers across the cortical depths is that layer positions and thicknesses vary systematically with the folding pattern. Here we can see that in gyri, the lower layers are expanded. In sulci, the upper layers are expanded. So if we consider layer 4, this granular layer shown in green, what this means is that layer 4 is sort of pulled up towards the peel surface in gyri and kind of pulled down towards the white matter surface in sulci. I like to think of this as sort of exaggerating the curvature of layer 4. So this has been seen classically from both von Economo and also from Bach, who went one step further to try to propose a mechanical principle that kind of explained this phenomenon that could potentially be used to estimate the layer positions for macroscopically observable characteristics such as the cortical curvature. And so the Leipzig group really went and implemented this equivolume principle to try to predict layer positions from the cortical curvature. So they started off with a classic Laplace solution and then tried to estimate the position, in this case, of layer 4 and compared the predicted location of layer 4 to the detected location that they could see in their high-resolution ex vivo data. We can see that the Laplace solution kind of minimizes the curvature of layer 4. As I said in the data, we see that the curvature is sort of exaggerated in layer 4. And it looks like the Laplace solution doesn't do such a good job in predicting the location of layer 4. They then looked at the sort of so-called equidistant approach, which assumes that layer 4 is at a fixed relative cortical depth across the folds. This did a better job. It didn't quite capture layer 4 perfectly. Again, this doesn't take into account this effect that I showed you in the previous slide. It doesn't vary with the folding pattern geometry. And finally, of course, they predicted the location of layer 4 using the equivolume principle. What they found was that this anatomically informed or inspired equivolume estimation performed best. And in fact, it's this approach that's used today in the majority of laminar fMRI studies for estimating the positions of the cortical layers. Now, this group went on in a subsequent study to note that we expect that the accuracy of the equivolume approach is expected to be greatest for small voxels. As we go to larger and larger voxels, the difference between the equivolume approach and equidistant approach may be minimal. So here's a comparison of the equidistant depth estimation and the equivolume depth estimation, animation provided to me by Renzo. Thanks, Renzo. So here you can see toggling back and forth between the two. As expected, the difference between these two depth estimations are more pronounced in places with high curvature, and they're pretty much the same in regions where the cortex is locally flat. So here's another example from Renzo looking at functional layer profiles using both the equivolume depth estimation and the equidistant depth estimation, showing that using the same kind of sub-millimeter CBV data that he mentioned in the previous slide. You can see in this case that the cortical depth profiles from these two depth estimations seem to be pretty much the same. And again, so what this shows us is that even with today's voxel sizes, the difference between the equivolume approach and equidistant approach doesn't seem to be very pronounced in our data. However, we do expect that as we go to smaller and smaller voxel sizes, we do expect the equivolume approach to perform better. There's other issues, complications in terms of estimating the positions of the layers. We know that there can be discontinuities in layers between adjacent cortical areas. The layer positions really jump around a lot across the cortex. You can see even between V1 and V2, we can see in the cell density in the myelin. So the mapping between cortical depth and cortical layer can vary a lot between neighboring cortical areas. So if you're performing a cortical laminar analysis in one cortical region, this might not be so important. But when you're trying to compare activation across multiple cortical areas, this is an important effect to take into account. Here's another example of this, showing varying thickness of the cortical layers across areas. Here I've picked a couple of primary sensory and motor regions. Of course, we know there's a lot of differences across the cortex. For example, there's no layer 4 and M1. We see that the layers really shift around quite a lot across regions, even between adjacent regions such as V1 and V2. Note also here that the thickest layers are around 500 microns. So what that means is that in order to comfortably resolve activation within an individual layer, we would need a voxel size of about 200 microns or so. So what this means is that in laminar fMRI, we very rarely, if ever, resolve the layers. So if I normalize these by the total cortical thickness, again, we can see how much the relative contribution of individual layers varies across the brain. We can see how huge layer 4 is, relatively speaking, in V1 compared to even V2. So this can also be a cause of sampling bias in fMRI. We're going to get a lot more signal from layer 4 when we're looking in V1, and therefore we need to take this into account in our analysis. So another approach to kind of estimate the positions of layers is a nice approach proposed by Peter Koopmans, in which he sort of aligned the cortical profiles directly to the anatomical data. So if he looked at the original profiles in the baseline fMRI data, he saw that there was no distinct characteristics across depth. He also saw there was a lot of variability of the cortical profiles across the ROI. So what he did was he sort of slid the profiles around a little bit and adjusted the cortical depths in order to make the profiles more consistent across the ROI. And in this case, he saw this nice little decrease in the baseline fMRI signal around the location of the striae of Gennari. And this allowed them to see this increased activation within layer 4 in response to visual stimuli. So I really like this approach. It adapts the layer positioning to a detectable laminar feature. It doesn't require an estimation or prediction of the layer positions. Now we need to be careful to not apply this approach directly to the fMRI data itself. This would be a little bit circular. But I really like the idea of aligning the depth profiles to some sort of detectable anatomical feature within the data. So we can also perform what we called anatomically informed smoothing. With this framework of small isotropic voxels and a family of intercortical surfaces, we can sort of embrace the full three-dimensional structure of the cortex. We don't have to approximate it as a 2D sheet, and we can perform kind of an anatomically restricted smoothing. And of course, we can position these intercortical surfaces however you like, based on equidistant or equivolume estimation. So for example, we can perform smoothing within cortical layers if we assume that the activation is constant within the cortical layers. We can also smooth across cortical columns if we have a prior that the activation will be columnar. We can also perform more general, what we call intercortical smoothing, again, with this 3D smoothing kernel that tracks the folds of the cortex, or what we call a restricted kernel, which sort of avoids contamination from the white matter and noise contamination from the CSF. I just want to point out that this tool is now a part of FreeSurfer, so I encourage you to give it a try. So here are some examples of performing this intercortical or interlaminar smoothing. Again, a very strong prior on the data in order to smooth within cortical depths. Again, data from Renzo. Here we can see in the original activation, you can kind of see that there's like 2 peaks in the fMRI data across the cortical depths. This becomes much more salient after he performed this kind of laminar-specific smoothing. And here's another example, again, where you can really see that the laminar-specific smoothing really kind of pulls out this feature in the data. But I like this image because it brings up a very important point, and that is when we're smoothing, we're kind of imposing a prior on the data. It's really important to make sure that if you ever do smooth, to look at the data both before and after smoothing, to make sure that the smoothing doesn't introduce any features into the data, and that it's really just a way to visualize a pattern that you can see in the raw data. Okay, so last I'd like to talk about cortical depth dependence of the hemodynamic response function. And as I've said, in order to interpret the fMRI signals, we need to take into account how the vasculature and the hemodynamics vary across the cortical depths. It's been shown that the bold response amplitude and shape vary systematically across cortical depths. This is because of the spatial gradient of microvascular dilation delays. So this is nice work from Anna DeVore's group looking at mouse cortex. You can see in this red ROI corresponding to the red trace shown on the right, the bold response is a little bit larger. Also note that it's got a very pronounced post-stimulus undershoot, and it also appears to have a little bit of initial dip in this red contour. If you compare that to the black contour, which samples from the parenchyma, there we see basically no post-stimulus undershoot, no dip, smaller response. So both the amplitudes and shape vary systematically. What this means is that any fMRI analysis using the same sort of canonical hemodynamic response function across all depths will also likely introduce the detection bias. You're more likely to see activation in the layer that has the hemodynamic response that matches your model. Now, this has been seen in humans as well, varying HRF features across critical depths. This is work from Jörn Sierro from the Utrecht group. Again, you can see very similar characteristics to what Anna showed in the mouse, including a pronounced post-stimulus undershoot and potentially an initial dip from signals measured near to the surface, and these features are absent from voxel sampling from the parenchyma. So again, depending on which layer you're looking at, fMRI analysis using one single hemodynamic response function will introduce some detection bias. And lastly, so even if there is local blood flow control that is layer specific, we still need to contend with the known downstream hemodynamic effects, especially in bold fMRI. The idea is that deoxygenated blood sort of drains upwards through the cortex on its way out of the brain. And what that means is that bold responses in upper layers are going to reflect an integrated response from all the lower layers. So if you consider, for example, if there's neural activity in layer 6, we'd expect to see some local bold activation in layer 6. But as the blood drains upwards towards the surface, we're also going to see activation in layer 4. This becomes even more complicated if we have activation also in the top of layer 4, neuronal activation. We're going to see some accumulation of the bold effects in layers 2, 3. So what you can see is that not only is there an accumulation of the bold signal across cortical depths, but it's going to be really hard to infer which cortical layers there's the neural activation just by looking at the pattern of bold signal across depths. So the Donders group, David, tried to model these so-called laminar spread functions in order to account for where we'd expect to see bold signal if there's neural activation in any given layer. You can see that these modeled laminar spread functions are highly asymmetric, which accounts for this accumulation of the bold signal. Now note that here I'm really considering these downstream hemodynamic effects as they pertain to bold fMRI. But because of the interconnected intracortical vasculature, we'd expect to see similar couplings, maybe more complex couplings of activation in non-bold fMRI signals as well. Okay, so just to give some conclusions and final thoughts, I've shown you how new preprocessing approaches can avoid inadvertent spatial smoothing and respect intracortical anatomy. I've shown you there's a lot of complexities in the mapping between cortical depth and cortical layer that needs to be taken into account in these analyses because layer positions shift across the folding pattern and even dramatically across cortical areas, even adjacent cortical areas. I've shown you how the hemodynamic response varies across cortical depth, which is another source of detection bias, and is coupled asymmetrically between the layers. I think that I will say that I think there's more need for tools for this analysis that are integrated into common software packages. I think many people who do laminar fMRI kind of have a lot of home-written tools that they use for themselves. I think that we need to have more sharing of these tools and some standardization. And so, final thought, and that is, in principle, laminar fMRI is easy, right? All we have to do is sample the signal across depths and see what we find. But I think that the challenges in laminar fMRI is the interpretation. The differences in the vascular responses across layers, plus this coupling that I mentioned, makes the interpretation really challenging. It's really hard to infer the underlying pattern of neural activity from these laminar fMRI signals. But I do think that if we can address these challenges, I think that laminar fMRI has a lot of potential for giving us sort of new insights, new fundamental insights into human brain circuitry and function. With that, I'd just like to thank a few people for contributing materials, especially Anna, Olivia, Villa, Shaheen, Natalia. Of course, thanks Renzo for the cool animations. Thank our funding sources for support, and thank you all for your attention.